The overall goal of this procedure is to drive the quantitative differentiation of T cells from mouse embryonic stem cells, or ESCs, in vitro. This is accomplished by first seeding the ESCs onto a monolayer of OP9 stromal cells. After 5 to 7 days, the mesodermal-like colony formations are harvested and replated onto a fresh OP9 cell monolayer. 3 days later, the small round hematopoietic progenitor cells, or HPCs, are harvested and transferred to a fresh OP9 monolayer for the generation of erythroid, monocytic, and B cells, or to a fresh OP9 DL1 monolayer to generate T cells. Ultimately, this co-culture method can be used as an in vitro model for studying the molecular, genetic, and cellular events that regulate hematopoiesis and T lymphocyte development.
This procedure was developed in the lab of Dr. Juan Zúñiga-Pflücker at the University of Toronto. We learned the technique from his group so that we could apply it to our research on gene regulation during T-cell development at Hunter College.
The main advantage of this technique over existing methods is that it allows for the experimental manipulation and observation of T-cell development using cell lines without the use of animals.
Begin by diluting 50,000 mouse ESCs in 10 milliliters of OP9 media per plate. Then, after removing the old media from the OP9 cultures, plate the ESC suspension, taking care to distribute the cells evenly, and incubate the cells at 37 degrees Celsius.